All right, chapter 4, focusing on our DNA, what else we do with our DNA, looking at how we make proteins, as well as how we make new cells. So let's focus first on DNA. So DNA is called deoxyribonucleic acid. It's very long polymer of nucleotides. We have 23 pairs of strands of DNA in our body. So 46 in total. So we got 23 from one parent, 23 from another parent. So our nucleotide monomers that create our DNA are made up of a sugar, a phosphate group, and a nitrogenous base. So for DNA specifically is deoxyribose sugar versus RNA, which is just ribose sugar. So there's just our pieced together parts of our nucleotide, in this case a DNA nucleotide because it's deoxyribose sugar. So deoxyribose sugar, a phosphate group, and in that case our nitrogenous base. So between our nucleotides, what's different is that nitrogenous base. So that's going to determine what type of nucleotide is the nucleotide. So for our nitrogenous bases, we have two sets. We have our purines and our pyrimidines. The purines are adenine and guanine. The pyrimidines are cytosine and thymine. In our RNA, the thymine is replaced by uracil, and again, only in RNA. So these are your bases. They're letter coded, based off of adenine A, guanine G, cytosine C, thymine T, or uracil for U. So there's your switch for your RNA. So both have adenine, both have cytosine, both have guanine. It's just that the difference is there's thymine in DNA, uracil in RNA. So the shape of DNA is it's called a double helix structure. So it's two ladders of our nucleotide polymerized to create a very long strand. We have some hydrogen bonds in between our pairing of nucleotides, and then it's all in a twisted helix shape. So our connectors between our two ladders are our pair of nitrogenous bases. So between these hydrogen bonds, we have the same exact pairing no matter where you are in your DNA strand. So an adenine is always going to pair with a thymine. A cytosine is always going to pair with a guanine. And this is called the law of complementary base pairing. So where one strand of DNA is going to determine the sequence of the other one. So if I did not have this side strand of DNA, I would automatically know that the partner for this one is a thymine, partner here, cytosine, partner here is another adenine, and partner here is a cytosine. So you always know who the partner is because it's always going to be the same. Okay, so one thing that you will do on your exam is to look at a original DNA sequence and tell me what the copy of that or your base pairing would be. So we have one-sided strand of our DNA, and then you're going to copy what the next one is. So it's all multiple choice though. So the partner DNA, we have our paired copies. And then we also have to do it for RNA. So from this case, wherever there was a thymine in the original one, there's now a uracil. So same pairing, except for when you get to a thymine, it's a uracil. So with our DNA, it has a lot of packaging in order to keep it organized. So there are these proteins called histones where the DNA wraps around a little bit, and that helps it to have little connection points, creating big folds and loops in order to create your organized DNA. So our levels of compaction, we have our original DNA strand. Then we have a nucleosome, which is a DNA wrapped around a little piece of histone protein, and there's a little bit of DNA in between it. Then our next level is chromatin, where it's a bunch of loops of these zigzaggy nucleosomes. Next level is chromatid, where, and you'll sometimes hear it referred as a chromosome, but it's that kind of X-shaped DNA compaction that you actually can see under a light microscope. So when a cell is not dividing, chromatin is the highest level compaction in a non-dividing cell. So this is still open enough where if you need to get to a particular gene in order to copy the RNA to get that protein blueprint, you can. Here, it's way too compact. It's just for making sure to get that DNA into the next cell. Okay, so this is another structural view of your chromatid. Okay, RNA. So RNA, a lot smaller than DNA. It's just either the copy of the gene or our other two types of RNA for our protein production. So we have three different types of RNA. Our messenger RNA, which is our gene copy. Ribosomal RNA, which is also called a ribosome. So the organelles you learned about last chapter. And then transferred RNA, which help with bringing in the correct amino acids to the coded messenger RNA, so making sure that order stays the way it should. Another difference between RNA and DNA, it's one nucleotide change instead of a double helix, ribose sugar instead of deoxyribose sugar, uracil replaces thymine as one of the bases, and the job of the RNA is mostly in your cytoplasm. Okay, so let's look at the genes and how to make proteins. So as a gene is a segment of DNA, it includes the instructions for making a protein, so a particular blueprint for that protein. So in our genome, which is all the genes of a person, only about 2% of it is actually coding for a protein. The other 98% has to deal with your structure of your chromosome, regulating whether or not a gene could be turned on or off. So making sure your protein production is highly regulated, making sure that you're not making copies of a gene that you don't need, telling your structures, hey, this is where the start of the gene is, this is where the stop of the gene is. So not all of it's actually coding for a protein. So I'll skip that. Okay, so for our gene, our copy of our RNA, the genetic code, so 3 nucleotides called a codon, is what's going to code for these particular amino acids. So it's going to tell the correct order for making of your protein. So again, our codon, our 3 base pair. So with our 4 different nucleotides, there are 64 possible codons. Some actually code for amino acids, so it's 61 of them. 3 of them are stop codons. So those are our stop codons. You don't have to know that for my test, but just knowing that there is a stop codon. The start codon is AUG. It codes for methionine, and that's going to be the starting amino acid for every single protein that you made. It might get cut off, it might not. It just depends on the protein production. So there is a repeating TA sequence called a TATA sequence that starts off, and then you hit that AUG, time to start making protein. Okay, so let's make a protein. So when we have a gene that is activated, as in able to be copied for making of our messenger RNA, we go to the first step, which is transcription. So sort of like, you know how you go to a doctor, the doctor writes you a prescription. So our DNA is kind of like all of the information that that doctor has in his head or her head or their head. All that information, but you only need something specific for your, infection or whatever you need a prescription for. So get that prescription. That occurs in the nucleus or doctor's office. And then you go out somewhere else. So in this case, for protein production, we're going out into the cytoplasm. That mRNA, our messenger RNA, is going to code for a protein. So this process is called translation. So translation as in your pharmacist takes the prescription that your doctor wrote, translate it into actual English versus scribble scrabble that it puts on the prescription, even though now they do more like typing stuff out. But translate that, get the correct medicine that you need. So an important enzyme in the transcription process, so going from DNA to RNA, so this is our first step, getting our copy of our gene, is RNA polymerase. So RNA enzyme to polymerize, so ACE for enzyme, to polymerize or put together the correct nucleotides to make RNA. So same thing we talked about base pairing earlier. It's going to read your original DNA strand, and put the correct partner, remembering where it would have put a thymine. If it was DNA, it puts a uracil instead. Okay, don't really have to know that part. You do have an initial mRNA that has some parts that are going to be translated, some parts that's not. Exons are going to be exited out, translated into a protein. Again, just part of gene regulation, making sure you're making the correct proteins that you need. Introns are going to be removed, not going to be translated into a protein. So if we have our original gene, go through transcription, just copy everything, and then we can splice. So even though we have a certain number of genes, we can actually make a whole lot more proteins depending on how we organize the exons in our pre-mRNA strand. So going from our pre-mRNA to mRNA, determining what sections we need to make whichever specific protein we need. So actually one gene can code for more than one protein. So next step is translation. 3 main participants. We have our mRNA, our blueprint for making of our protein. It has our codons, our 3 nucleotides in order to code for a specific amino acid. The next one is the ribosome, so our big chunky purple thing here. Where are we? There it is. So that is going to hold everything together, our nice scaffold. It's either going to be free-flowing in the cytoplasm or attached to our rough endoplasmic reticulum. So those are for proteins that need to be packaged into a vesicle. The last one, our cute little curly Q's up here, those are transfer RNA. So they are bound to a amino acid, and when it's their time, when this anticodon, the bottom part of the tRNA, matches with a codon on our mRNA, it's going to bring in the correct amino acid. A peptide bond is formed, and then we shift, move to the next codon, and then able to bring in a new tRNA, make another peptide bond, and keep elongating our I just went blank. Amino acid chain so we can make our protein. Okay, so translation, we start off again our AUG, our methionine, as our first first codon, bringing in the first amino acid, and then this ribosome is going to shift and move to the next one. So everyone else stays in place. The ribosome shifts so that moves everything over, makes this open spot, new tRNA brings in another amino acid. The one that leaves, this exit row, can go form another bond. So it takes energy in order to do this, so you're using up ATP to create that bond and waiting for its turn to bring another amino acid. So here's just your tRNA, your transfer RNA, the anticodon that's going to pair up with the codon on the messenger RNA, and this end section is where the amino acid is. Okay. I'm going to skim through this because I don't think that's on the test as much. So you do have your first phase where you have your mRNA, your ribosome binds to it, finds that AUG codon to start with, and then you start bringing in other tRNAs. That's a level of detail that I know is not on my test. Okay, so you keep going, you keep bringing in more amino acids, adding them to your chain, and eventually you're going to reach to a stop codon. A stop codon doesn't code for an amino acid. So when that happens, it just releases. So if it is a ribosome that's free-flowing in your cytoplasm, then the protein just breaks off and you're good to go. But if you are a vesicle-enclosed protein, you're actually packaged into a vesicle, which is on a different slide. Oops. Okay, so just showing translation. All the little green is your growing amino acid chain. These little bluish blobs, that's actually the ribosomes on our pink mRNA. So your translation, you can have a ton of proteins being made off that same piece of mRNA. Okay, so vesicle-enclosed protein. So here we have our ribosome attached to our rough endoplasmic reticulum. And as the amino acids are being put together with peptide bonds, they're being fed into our rough ER, eventually creating a transport vesicle using the phospholipids of the rough ER. And then this transport vesicle actually is going to make another stop before it has to do stuff. So it's actually going to go to the Golgi complex, which is on a different slide. Okay, so review peptide formation. 2 different steps so you can practice that. Skip that. Okay, Golgi. So your Golgi is going to be your final step. So we have our vesicle for our vesicle-enclosed proteins. They're going to merge with the membrane of our Golgi complex. The Golgi is going to modify the structure, make sure everything's organized right. Sometimes put carbohydrate chains on it. So it's your final step or stop for protein modification. And then that final vesicle buds off, and then you're able to go through exocytosis, or if it's something that's just being inserted into the membrane, then it's just inserted into your plasma membrane, just stuck there. So even though you have endocytosis, which can use up some of your plasma membrane to create those vesicles, when you have the release of vesicles, either releasing the product or inserting something into a plasma membrane, it actually adds to the plasma membrane. So it's not like you can keep eating up your plasma membrane and then you're kind of, oops, I don't have any more left. There's a adding and a subtracting that keeps going back and forth. So with your proteins, in order to make sure that they are functioning correctly, you do have these what's called chaperone proteins, making sure they're folding correctly, making sure that, for example, with hemoglobin, you have multiple proteins that create hemoglobin, so making sure that all of your subunits that create it are coming together, forming the correct bonds. There's another way to call these. They're called stress proteins or heat shock proteins. So sometimes if there is a little bit of a damage, these chaperone proteins can kind of come in and fix stuff, put it back in the correct shape, just so you don't have to remake that protein all over again. Okay, so differences in your proteins, either on free ribosomes and your cytosol, rough ER, for packaging into a vesicle, and then sent to the Golgi for your final modifications. Keep going. Okay, genes can be turned on and off, just depends on what you need. Sometimes your other stuff that is made in your body, like glycogen, storage of glucose, making of your triglycerides, which is your fat, making other stuff, even though there isn't a gene for it, there's usually a gene for a protein that's the enzyme that you need in order to do that. And so your product kind of has some gene control, but not directly. Okay, I'm going to start a new video on this one.